Okay, so good evening, everyone. This is week two of your NPTEL live session. So we'll wait for 5 minutes for other people to join, and then we'll start the session. So we'll just wait for 5 minutes so that uh, other people can also join, and then we'll start the session. Okay, so I guess we can uh, start with the session. And because last session as well as we started with the session, people kept on joining. So those who are late, they can join later as well. So for your week two of NPTEL live session, I hope I am audible to everyone and my screen is visible to everyone, right? Can you please like write in the chat box if I'm audible to everyone? Okay, fine. Thank you. So we'll start with the session. So in this session, based on your course content that was uploaded on NPTEL website. Okay, so I guess people are still joining. I request everyone if you guys can like join the session on time, because what happens is it will help me to, you know, start the session on time and we have enough time to have proper scientific discussions. You guys, if you guys have any kind of doubt related to some topics or not just related to topic, if you have any other doubt related to, you know, any other thing, or you have any kind of curiosity, any questions in your mind related to nanotechnology, how things work, no matter you know, even if it's a very basic doubt, but I will be happy to address that. So if you guys will join on time, it will be easy for me to let everyone in at the, because people are still joining, so it will be easy for me to let people in on time and start the session on time. So I guess people are still joining. We'll have to wait for 2 more minutes so that they do not miss uh, any of the topics that we are discussing now. Just 2 more minutes. But before that, if you guys have any doubt related to previous session or have any questions related to this particular course, feel free to write that in the chat box and I would be happy to answer those. Okay. Okay, so as we were discussing in your week two of the session, okay, so in your week two of the session, course content that was uploaded on the NPTEL website, it was basically DNA nanotechnology. So basically, it deals with how you can use DNA, basically genetic material, for various nanotechnology applications. It could vary, like it could be used for biosensing or sensing applications. It could be used for, you know, therapeutics. It could be used for diagnostics. It could be used for detecting diseases by framing some lateral flow assay devices or like that. And apart from that, how proteins or maybe glycoproteins or that or lipid can be used in various nanotechnology based applications. And another interesting thing was these bio nano machines. So what was there in the bio nano machines? It was basically how we can make use of the uh, bio inspired designs or bio inspired nano machines that are already functioning in our body for different biomedical applications. And the last topic was carbon nanotubes and its applications, how carbon nanotubes are synthesized and what are its various applications, how we can, engineer carbon nanotubes basically to for different biomedical applications. So we'll move forward. So what exactly is DNA nanotechnology? So nanotechnology is a field that uses synthetic DNA and nucleic acids as building blocks to design and construct nanoscale devices and materials with applications in various technologies. So what does it do? It exploits DNA's predictable, programmable, self-assembly properties and its ability to organize other functional materials rather than its own role as genetic information carrier. So what is happening here? Just a second. So what is happening here? DNA has a property of this self-assembly property. So you can easily program, you can easily tune DNA for different applications by synthesizing nanostructures out of it. So its ability to organize other functional materials. So it's not just DNA has this property because we know that it follows Watson-Crick pairing. It can self-assemble, but not just that. It can also induce other materials, other functional materials to align in a proper fashion. So rather than its role, so here DNA is being used not as a genetic material. It is not used as a genetic, not just genetic information carrier, but also it is being synthesized, it is being engineered into a nanostructure which can be used for different biomedical applications or different nanotechnology based applications. So what are some of the key applications? Some of the key applications include the creation of biosensors. That is one of it. We'll see some of the examples. We'll see some of the schematics in which we'll see what exactly is happening, which can be used for disease detection, drug delivery systems, and templates for creating larger structure, right? So these are some of the basic applications in which DNA nanotechnology is currently being used. Moving forward, DNA nanotechnology uses the natural base pairing rules of DNA. We all know Watson-Crick pairing rule. Like we know that A adenine forms double bond with thymine, so basically purines and pyrimidines, and guanosine forms three hydrogen bonds with cytosine. So DNA nanotechnology uses this basic principle only. It exploits this basic principle of natural base pairing rules to, to do what? To build nanoscale structures and hybrid materials. Instead of just storing genetic information, DNA becomes a programmable material for engineering. So this particular DNA nanotechnology, if we are talking about the different applications and how exactly we are employing it for different purposes, so it has been broadly divided into two sections. So this is just I have summarized in this picture what exactly we'll be discussing. So we'll be discussing all the applications, what is happening. So DNA nanotechnology can be broadly classified in two broad categories. The first one is self-assembled DNA structures in which DNA is being self-assembled. Okay. And the other one is inorganic materials, how DNA is being combined with inorganic materials. Okay. How DNA is incorporated how DNA is incorporated with different inorganic materials and how that particular conjugation, how that particular incorporation of DNA with inorganic materials are being exploited or are being used for different biomedical applications, for different, for drug delivery, how it is improving the targeting capability of those particular nanoparticles or nanostructures to which DNA is being, attached. Now we'll see all of those. Now, what exactly are self-assembled DNA structures? So this, although obviously you guys have a lot of course material already available for these topics that we are discussing, and sir has also discussed in his video, like what exactly is DNA origami. But DNA origami, I guess, is a little new concept for all of you. So I decided we will just have a look at this topic in a little brief so that you guys understand it better. People are still joining. I still request everyone, if you guys can join, on time, we'll be able to. That would be a smooth conduct of the classes, but it's okay. And at any point, if you guys feel that I am not audible or the screen is not visible, please write in the chat box. Or if in the meantime, while we are discussing all these topics, and you have any doubt, you can instantly write your doubts in the chat box. I'll be addressing your queries. So self-assembled, what are self-assembled DNA structures? So DNA itself is the raw material. Keep this in mind. We have just now seen that DNA nanotechnology, we divided it into two different categories, two broad categories. The first one was in which DNA itself is used as the raw material and there is self-assembly happening. Okay, you are inducing DNA to take certain structures. So here DNA itself is the raw material. So, by designing specific sequences, we can fold or link DNA into different nanoscale structures. Okay, so how is it happening? So, long single stranded DNA. Suppose we have a scaffold or long single stranded DNA that is already there. Now, this particular scaffold, this particular long DNA structures will have some sequences, right? It will have sequences here. So, this is a long scaffold that we have. Now, what happens next in this particular DNA origami thing, how it works, how different nanoscale structures are created. So what happens is, apart from this particular long scaffold, please let me write it so that it is easy for you guys to understand even after you, even during the time when you see the video on uploaded video on YouTube. So this is a scaffold, okay. So apart from this, there would be some staple strands of DNA that will be taken. Now, the characteristic for these staple strands of DNA is this particular staple strand will have complementarity with some of the regions present here in the scaffold. So this particular DNA staple strand will come and bind here and get hybridized. So different staple strands are there. So different staple strands are there which will bind to different regions in this scaffold, and it will take a certain structure. Suppose this, it will take some kind of structures like this or like this, and we can verify it by, so these are the AFM images. If you guys remember, we have discussed what is AFM. What is AFM? AFM is atomic force microscopy. So what was happening in atomic force microscopy? Let's recall what was happening. So there was a, suppose this is the glass slide or the where you are keeping your sample. You casted your sample here. You suppose you casted your sample here, and suppose you casted your sample here. So there is a cantilever along with a tip. So this particular tip, it moves through your sample in a raster scan mode. What is raster scan mode? It will move like this. In a raster scan mode, it moves through your sample, and based on this, so there is this laser coming. So whenever this particular tip interacts with your sample, so this cantilever, this bendable cantilever bends a little, and uh, based on the amount of bending, whatever information is extracted, you get to know what exactly is the structure. So this was the whole concept of AFM. I hope you guys remember. And if you guys, if some of you have joined this session but missed previous session, I would suggest to go on the YouTube link that was given to you and see that video in which we have discussed different techniques like atomic force microscopy, zeta potential analysis, dynamic light scattering. All these are very important techniques that you should know if you are studying nanotechnology, if you are dealing with nanoparticles. because these are the characterization techniques which are very important for you to know the basic, like it's very important for you guys to know its basic principles, because these are the main characterization techniques to see if your nanoparticles are being formed, what are your nanostructures, and how does it look for morphological and for various, basically for various morphological and functional characterization. You guys should know this. Now, coming again to the DNA origami. So, long single-stranded DNA is folded into 2D or 3D nanostructures using short staple strands. Just now I explained you there is a strand, long DNA strand which we call scaffold, and some staple strands, small strands, which are particularly which have sequences that you have decided to go and bind at specific locations in this particular scaffold strand. And then based on that, this long DNA strand is folded into 2D or 3D nanostructures. The structures can look like boxes, tubes, or even robots. Applications. What are the applications? Smart drug carriers that release medicine at disease sites. Okay. Nanoscale machines for biosensing and template for nanofabrication. So we'll see all the applications in detail as we move forward in the session. So DNA origami is one of the most recent techniques of utilizing DNA as building blocks for synthesis of nanoparticles. It is one of the latest methods in the field of nanotechnology, having its own limitations and opportunities. Now, whenever we are talking about a particular approach, whenever we are talking about, any method, so there is always some advantages of it, and apart from that, there are also some limitations. Now, what are the limitations, particularly when we talk about DNA origami, is the number of folds that we can achieve, you know, the number of folds, how many times you're folding, what structure you're getting. So after a certain limit for extremely complex structures, you cannot use extremely complex structures, you cannot synthesize using this DNA origami. So there is a, I would say there is a cutout point uh, up till where you can use DNA origami for synthesizing different nanostructures. But after that, if the structure is extremely complex, if there are so many foldings required, so many staple stands to bring out a complex structure, then at that point it would fail. Now, uh, again, this one is origami is a Japanese word that means folding of plain sheet into an arbitrary form having a specific dimension. Long strands of DNA are folded into a complex scaffold of staple strands having 200 to 300 nucleotides. This leads to formation of a complex structure that has characteristic feature because of their nanoscale dimension. Now, let us have a look at the history of DNA origami. So this one is important. So the field of structural DNA nanotechnology was born in 1980s with initial work by Seaman, who proposed building geometric structures with multiple strands of DNA at a nanoscale level. However, complications in synthesis and stoichiometric control. Now, pay attention to this particular term, stoichiometric control. It is very important to regulate this stoichiometric control. What does it mean? It means the scaffold that you are taking, the long scaffold that you are taking, you're starting with, and your staple strands. So you need to optimize the ratio in which you are taking it and the amount that are there. So stoichiometric control left the field lacking in more promising developments. In 2004, Joyce group demonstrated that a long single-strand DNA could be folded into a geometric shape, octahedron, with the help of synthetic shorter oligomers. Okay. Taking this further, Paul Rothemuth in landmark discovery demonstrated that long single-stranded DNA could be folded at nanoscale levels with the help of smaller staple strands to create various shapes. Specifically, Rothemuth demonstrated the creation of complex single-layered nanostructures that were 100 nanometers in diameter and had a spatial resolution of 6 nanometers. You can imagine such a great spatial resolution. So he termed it as DNA origami. This approach creates custom nanoscale shapes that are automatically precise by taking advantage of specificity of Watson-Crick pairing. I mentioned it again. So, what important points you need to, remember are that first of all, you should always know that whenever we are talking, whenever we are developing a new methodology, whenever we are developing a new approach, new application, it doesn't happen in a flick of a second. So, it's not like a breakthrough researches just happens in, that one day you started doing some experiment and you got the results. So it's always like somebody proposed something, and there's always addition from time to time. As we move forward, people do some research on that, improves that method. Then after some years again, there would be people working on it who would further improve the that particular approach. So research progresses like this. It doesn't happen in just one day. It's a very long process and it keeps on improving. So two important things that we can you know interpret from this history is that people found out that they can use a long strand of DNA, and by employing Watson-Crick base pairing and some staple strands, they can synthesize nanoscale structures that were 100 nanometer in diameter, or maybe they can synthesize basically nanoscale structures. So this was the whole point. Now let's understand the process, that how is it happening. Let's understand how is it happening in detail. So DNA origami is a method where a long single-stranded DNA scaffold, often viral DNA like M13 bacteriophage DNA. If you remember, we have also seen virus-like particles and especially bacteriophage. How does it look? What is its morphology? And where bacteriophage is being used, like it is being used as a basically delivery carrier, as a vector for different drugs or bioactive molecules. So it is folded into specific nanoscale shapes with the help of many short synthetic DNA staple strands. So as we start with this process, this DNA origami, so first of all, we have to do selection of a scaffold strand. Okay. So first of all, you need to select a scaffold. Okay, based on what sequences you require. Based on what sequences you require. So a long single-stranded DNA is chosen, M13 bacteriophage with 7,000 bases. Okay, this scaffold acts as the backbone of the origami structure. So always remember, whenever you're talking about origami, this scaffold strand is its backbone. Now, next important and extremely critical step is design of staple strands, because ultimately it is this staple strands that you are giving that will lead to folding of this particular scaffold. Ultimately, it is the staple strands that is leading to, see, that is leading to folding of your, this is your scaffold, and these staple strands, these blue in color, these are ultimately causing folding of your staple strand and ultimately leading to formation of DME origami nanostructures. So based on different staples, based on different staple strands that you are using, you can synthesize different kind of nanoscale structures. Okay. Just a second. So based on that, you can synthesize different nanoscale structures. So design of staple strands. Hundreds of short synthetic DNA strands, 2 to 60 bases, are designed using computer software. Okay. So again, how you synthesize, it's not like you just think of something and just come on the workbench and start synthesizing DNA strands based on specific sequences. No, this is not how it happens. First of all, there are different computational tools using which you design a particular synthetic DNA strand based on what kind of nanoscale structure you want to draw out of it, right? So, are designed using computer software. Each staple is complementary to two or more distinct regions of the scaffold. Now, one very important thing to keep in mind is that if a particular staple strand, suppose it is binding here, so you have to design the sequence of other staple strands so that it is binding at some distant locations. Because if it will bind here only, they would be overlapping, and the structure that you're trying to make may not be the one. Because so you need to see at what distance you are letting the staple strand bind to the scaffold DNA. This is a very important point. So each staple is complementary to two or more distant regions of the scaffold, so it can stitch parts of the scaffold together, right? Now moving forward, next step is you mix the scaffold and staples together. Scaffold DNA and staple strands are mixed in a buffer solution containing magnesium ions. Why magnesium ions? Because magnesium ions is crucial because it stabilizes the negatively charged DNA duplexes. So if you see here, what is happening is, suppose you mix the staple mix and scaffold together, and this kind of structure it forms. So this is your scaffold, this is your staple strand, right? This is your scaffold, this is your staple strand. So in this condition, if irradiate this particular with light, there would be low scattering. So basically it is, these experiments are done to predict if your DNA origami structures are formed or not. So once you add these staples and let the reaction proceed, and there is this nanostructure formed, so there would be some excess staples as well. There would be some excess staples in the solution as well, but there would be this DNA origami that you have just induced, just formed. So in this case, what would be the difference is, in this case, when you will shine light, there will be low scattering. But in this case, when you shine light, there would be high scattering. So this is how you detect also that your DNA origami has successfully been formed. Now, once you mix the scaffold DNA with the staple strands, uh, then you let the reaction set. After that, you do the denaturation, as you can see. This mixture is heated 90 to 95 degrees. This step separates secondary structures and ensures all DNA is single-stranded, ready for pairing. So if your DNA is like that, so first of all, you need to make it single-stranded. So that it can pair. Next is annealing, which is cooling plus hybridization. This mixture is slowly cooled back to room temperature, as you can see, from 65 to 40 degrees Celsius. This mixture is slowly cooled back to room temperature, and what happens during cooling is there is hybridization occurs. So first of all, you did separated the single strands, and then in the next process, when hybridize, you cool it down, and then you let the hybridization process. So this particular strand is there. It will, so you let the hybridization process go, and it will bind with your staple strands. So hybridization occurs, complementary base pairing. A binds with, basically adenine binds with thymine and guanosine binds with cytosine between staples and scaffold regions. Stepwise hybridization pulls different regions of the scaffold together. This folding process transforms the scaffold into the designed 2D or 3D shape. Right now, what is folding or self-assembly? The scaffold, held in place by dozens to hundreds of staples, completes its folding into the final origami nanostructure. Final origami nanostructure. So what is happening? Let's have a look here. Just let's have a look here. So we have the scaffold strand here. We have different staple strands here. You can see this is single-stranded DNA scaffold. These are staple strands. Okay. So after some time, after annealing, what would happen? These DNA origami nanostructures are formed. Okay. So once it is formed, once it is, this is final origami nanostructure. So the next step is purification. Why do we need purification? Because we need to remove these excess staple strands. So these are in excess. These are the staple strands that has not been used. These are used in excess basically. So these excess staple strands need to be removed. So excess staples are removed using techniques such as gel electrophoresis or filtration. This ensures only correctly folded DNA origami structures remain. Then we do, once we are, obviously, as you guys would remember that in the previous lecture we discussed that once our nanoparticles are formed, the next step is we characterize them. Basically, we check if they are formed or not. So we do the characterization by various techniques. Once the DNA origami structures are formed, we do the characterization, basically verification if they are formed. So the final structures are checked and visualized using atomic force microscopy. If you are forming a 2D surface, if you are visualizing the 2D surface, transmission electron microscopy for basically having to know the 3D dimensions, and gel electrophoresis basically to confirm correct folding patterns. Okay, somebody has shown uh, a downside. If you guys do not, I mean, you're not understanding something, you can like write in the chat box. If you, because see, whatever topics I'm teaching, these are basically your course related topics only. So it's not something that I have brought up on my own. So if you guys do not understand something, or if you guys feel that it is like getting monotonous after some time, you can tell me. We can talk about some different applications as well. So I just want the session to be lively so that you guys understand what exactly is happening, and a little in detail, because you have a lot of content in your on the videos that is uploaded on your NPTEL website. So I just want, so some important concepts, important theory that is there in those lectures, I'm just trying to revisit those theories so that you people understand it more effectively. So it's just that if at any certain point you guys are not understanding anything, or you feel like there is some problem, you can definitely tell me in the chat box. Okay. Make materials at the nanoscale. How do we actually control the quantum effects that start showing there? Because it feels like we can't really see them directly. Okay, so what exactly do you mean with the quantum effects? Because see, there are different things. If you are kind of so okay, I guess you need to tell me what kind of quantum effects you're talking about. Because if you remember in the previous lecture we discussed that if you are using a fluorescent dye, and if you are loading some kind of drug or bioactive molecules along with that fluorescent dye, there would be quantum, basically fluorescence quenching of that. So it's basically its fluorescence would be quenched because it is somehow interacting with the other molecules or the other bioactive molecules, because of which it is undergoing some kind of modification. So what exactly do you mean by the key, how we actually control the quantum effects? Because I guess a lot of things change, a lot of observations are there. Because I'll just explain you a simple thing. Okay, suppose you are making, let's not talk, because see, in case of gold nanoparticles, if you remember, we discussed how gold nanoparticles are formed, and how when we are making gold nanoparticles, depending on the amount of reducing agent we are adding. Suppose we are making gold nanoparticles. Okay, in all of it, in all of these tubes we have, just a second, क्लोरोरिक एसिड हमारे पास है, ये HAuCl4 हमारे पास है. नाउ हम इसमें reducing agent ऐड कर रहे हैं. Depending on the amount of reducing agent that we are adding, you will see different colors. Okay, so sometimes when you are adding low, it could be like red wine in color. And when the nanoparticles, the gold nanoparticles that are formed are bigger in size, it would some appear purple. Okay. So these are the kind of effects that you can see with your naked eye. And obviously you will have to do the characterization, you will have to record its absorption spectra, which will show a characteristic absorption maxima like at 520 to 530 nanometer of characteristic of your gold nanoparticles. But these are some of the cues, I would say. These are some of the cues, basically visible cues that you can see with your naked eyes. These are some of the visible cues that you can see. Now, as the question, based on the question that you asked, that how will we see what kind of quantum effects basically would be there and how to see. So these are the visible cues that we can see. But apart from that, there's sometimes the suppose you are using a protein-based polymeric materials that is transparent in color. Suppose this is your MCT. Okay, you took something here and you don't, and इन you took a polymer, ये आपका पॉलीमर है, सपोज ये आपका पॉलीमर है, ठीक. You added some kind of salts in it. Okay, you added some kind of salts in it. So how to know? Everything is transparent, this solution is transparent, this solution is transparent, but still you would notice some changes. I will just show you one thing. So there is one of my uh, papers. So I'll just search and show you, because there we have that. just a second. So you can actually see. So if you're talking about the visible cues, because as you mentioned that when we are working at nanoscale, we cannot see what exactly is happening, if it is being formed, what exactly is happening. So let us have a look at one of the figures. Okay, I'm zooming out this figure for you. Okay. Can you see what exactly is happening? I guess if you see, this solution is extremely transparent. I hope my screen is visible to everyone. Okay. I don't know what's wrong with internet this time. Okay, so I cannot zoom it more than that. But what we're doing is we are taking a polymer here and we are adding some salts in it. Okay, so it might not look very clear, but you can search it later as well. But if you look at this, it is extremely kind of transparent. This polyl lysine, basically, which is a polymer of lysine residues. When we are adding salts in it, you would see that it would somehow become a little hazy or opaque. So these are also the visible cues. So it's not like there would be some different color changes and like that where you will see basically what exactly is happening. So there are always some cues. Sometimes it could be visible. Sometimes it could be like you would see there is aggregation after addition of some sort of agent. Then also it is a positive indication that your nanoparticles are, because what happens is, suppose you have simple lysine residues. Okay, I'll again go back to your presentation. I'll again go back to your presentation and just explain what exactly is happening. So that's the whole concept of self-assembly. Suppose you have different lysine residues like this. Okay, now what you're doing is when you're adding salts to it, they are kind of aggregating like this. They are forming these spherical shapes. You are basically inducing self-assembly of these lysine polymers. Okay, so this is what you are doing. So when this happens now, so here you would see it as a transparent, but here what happens is you would see some kind of aggregation where it would turn opaque, it would turn hazy, and you would see that okay, after addition of these salts, there is some changes. So these are the some of the cues that you get when you are working, when you're working in synthesizing nanomaterials. So this is my take. But if you still have doubt, because you wrote, how do we control the different quantum effects? So what exactly you mean by quantum effects? If you can explain, I can like tell you in detail what exactly you mean or what you're talking about. So till then, this is the concept of self-assembly, because self-assembly, sir also taught you guys self-assembly in the lectures that he has uploaded. So self-assembly is an extremely important, I would say, uh, characteristic that is, I would say, method that is being exploited for synthesis of different nanoscale structures. DNA origami is based on self-assembly. Your synthesis of different polymeric materials, also different polymeric nanoparticles can also be synthesized using self-assembly. So this is the concept. Now moving forward, again, I have tried to visualize it here. There are different staple strands that you have, and based on this, you took one scaffold, one long scaffold. So these staple strands would push, basically pull your long scaffold and give it a particular shape. So this is a scaffolded origami. This is a multi-stranded, you whatever the staple strands that those are there. So building nanostructures by assembling multiple different DNA molecules becomes extremely difficult beyond a certain level of complexity. So this is the limitation. Always remember the limitation. The DNA origami approach greatly simplifies building DNA nanostructures by using one very long DNA strand and folding it up to form a scaffold. A large number of much shorter staple strands are added in excess to help folding. The staple strands bind at specific sites along the longer scaffolds. Now this is binding here, so it will bind at specific site along the longer scaffold to drive folding. This approach means that it is no longer necessary to strictly control the ratio of different DNA strands as for the traditional DNA folding described in previous section. Assembly, this self-assembly is much faster and yields are much higher with the origami approach. Now, people can also custom tuning, like just now I told you about gold nanoparticles, okay? So based on the amount of reducing agent you are adding to it, you can control the size of different gold nanoparticles that you're synthesizing. Suppose 20 nanometer, 50 nanometer, 100 nanometer. So based on your intended application, you can synthesize nanoparticles, basically metal nanoparticles or your polymeric nanoparticles of different sizes based on what intended application you have. So similarly for DNA origami structures as well, depending on the scaffold size, what scaffold size you are taking. Suppose 744 nucleotides, 1512 nucleotides, 2256 nucleotides. So as you would see that as we are increasing the number of nucleotides in a scaffold, the shape that we're getting is same, but its size is increasing. So that is why we term this, we can custom tune it. We can custom tune the size of nanostructures. Next one is, this one is very important. So this is an interesting concept as well in which this particular DNA origami scaffold production. So how is it being done? How DNA origami scaffold production is being done using bacteriophage based methods. So as we all know that bacteriophage basically is a virus that infects bacteria. Now what is happening here is these are simple M13MP. Suppose this is the specification. It's a bacteriophage. Now you took this bacteriophage and infected the E. coli cells. Okay, you let the cells multiply, grow, and in a bioreactor, and then you purify it. And in this case, if you're doing it, you will get single-stranded DNA. Okay, single-stranded DNA basically. But this is the modified approach. This B panel basically represents a modified approach in which, if you read it here, bacteriophage based methods for DNA origami scaffold production. The first one is the usual thing in which we are using this particular bacteriophage to infect E. coli bacteria, where it can replicate and form progeny phages that are released in the culture medium. Okay, extracted and purified to yield single strands of DNA scaffolds. These are single strands of DNA scaffolds. Now, how you can do this custom production of DNA origami? So, custom single-stranded scaffolds of different lengths and sequences based on what is your requirement, what length you require, what sequences you require. These custom single-stranded DNA scaffolds are encoded into phagemate. So this is a phagemate. What exactly is a phagemate? I guess you all would have listened about plasmid, but what exactly is a phagemate? Phagemate are cloning vectors that combine features of both plasmid and bacteriophages. So enabling them to replicate as a double-stranded plasmid, because as we all know that plasmid follows this replication pattern, and it also acts as a bacteriophage and be packaged as single-stranded DNA into a viral particle by a helper phage. So this phagemates act both as plasmid and bacteriophages. So this particular, suppose we require a particular length of scaffold and we require a particular sequence. So what we do, we took that and we encoded it in a phagemate. We encoded it in a phagemate along with other essential genes for replications like origin of replication. It should have origin of replication, obviously. The phagemate is used to transform the host E. coli in the presence of a helper phage. So one important consideration is that apart from this phagemate, there shall also be a helper phage. What would this helper phage do? It will basically provide structural, uh, I would say structural components to this phagemate for it to properly act. So the phagement is used to transform the host E. coli in the presence of a helper phage of a helper plasmid. The transformed infected host cells are grown and the extruded progeny phages containing target single-stranded DNA are extracted. So now what is the difference? What we need to see here in both these approaches is, here all of the single-stranded DNA scaffolds that we are getting are similar. Now here we can see These are custom single-stranded DNA. So they have different lengths, they have different size and different sequences as well. So based on your requirements, you can synthesize custom single-stranded DNA. So here again, we have discussed what exactly a phagemid is for your better understanding. So a phagemid is a plasmid that carries both plasmid origin of replication, allows normal plasmid replication in E. coli, and apart from that, it also contains F1 ORI, phage origin of replication. So it has origin of replication for both plasmid and for phage as well. Allows production of single-stranded DNA, which is needed as the scaffold in DNA origami. However, phagemids cannot make phage proteins on their own. So why do they need? Just now I told you that apart from the phagemid, it also needs a helper phage. It needs a helper phage. So when it needs a helper phage, what does helper phage do? Helper phage is a modified filamentous phage. It provides all phage structural and replication proteins. So apart, phagemid already has this plasmid origin of replication and phage origin of replication, but it does not have phage proteins, basically structural and replication proteins, which are required to package DNA into phage particles. So that is why we need a helper phage along with phagemid. The phagemate supplies the DNA to be packaged while the helper phage supplies the machinery. So it has DNA and it supplies the machinery. Basically, helper phage supplies the machinery. Result, phagemate derived single stranded DNA scaffolds is secreted into the culture medium ready for purification. Problem, what is the problem? Helper phage also packages its own genome. So what is the limitation of this? The limitation is sometimes the helper phage, obviously it is providing structure and replication proteins and basically machinery, but apart from packing this, it also packages some unwanted single stranded DNA. Now, role of helper plasmid. What does helper plasmid do? A helper plasmid is an alternative to helper phage. It contains genes for phage proteins. Therefore, it provides the necessary proteins, but only the phagemate DNA is packaged. So this one is interesting. In this case, यहाँ पे there was contamination because of the own genome of the helper phage. But in this case, it only supplies the helper plasmid only supplies structural proteins, and there is only this ensures high purity of scaffold DNA, no helper DNA contamination. So basically, what I wanted to teach you, these approaches, which are, well, I mean, which just looks like simple schematic diagrams, actually they are very complex. These methodologies are very labor intensive, and you have to put a lot of planning into it, that how things are being controlled. But yes, the approaches are very interesting. How you're using a phagemid and using it, to make different custom single stranded DNA support. So this is the, I would say, the beauty of molecular biology, how you can basically explore it for different applications. Now, another thing in which DNA based nanotechnology is being used is we make DNA hydrogels. So these are networks of DNA strands cross-linked to form gel-like materials. So here also DNA is being used and it is being cross-linked with some materials to form gel-like materials. They can hold large amounts of water and respond to stimuli, pH, temperature, enzymes. What are the applications? The applications include controlled release of drugs or growth factors. So suppose you make a hydrogel by cross-linking it with something, and in this hydrogel, what you did, you loaded some bioactive molecules. It could be drugs, it could be dye, it could be any other thing that you wanted to load. Now suppose you make a hydrogel and you loaded your drug in it. Now it is properly packaged. Now what happens next is, suppose in present of different internal or external stimuli, suppose there is change in pH. Suppose this particular hydrogel is stable at physiological pH, but when it goes to tumor microenvironment, suppose if the pH at your target site is less than pH, basically is less than suppose 5 acidic pH hai. If it has acidic pH, so this particular hydrogel is breaking I mean, it's releasing, just a second, it is releasing the target molecules there. So you can tune like how do you want, under what factors, under what conditions you want that this particular hydrogel releases the bioactive molecules in the surrounding. So all these are kind of approaches that are used for different drug delivery applications, for delivering chemotherapeutic drugs, for delivering, basically the bioactives for gene therapy. So DNA hydrogels is one such application. Now the next one is signal amplification system. DNA can be engineered to replicate or amplify itself in response to a target. Example, rolling circle amplification hybridization chain reaction. This creates a strong signal from a tiny amount of input. So, signal amplification system. So, let us understand this particular application of DNA nanotechnology in a very simple terms. Suppose you took a DNA scaffold, DNA staples, and made a particular DNA origami nanostructure. I guess it's clear till this particular step. Now the next thing you did, you attach some of the probe molecules, suppose some of the fluorescent tags or gold nanoparticles or something, which are specific to detecting some other molecules. Now these tags that you have added, these are specific to detecting some other molecules, and these are fluorophore probes, so they would show some kind of, you know, color change or some kind of changes. Now, so basically, these are being used, this particular approach in which we use DNA origami nanostructure for lateral flow simple signal amplification. We make lateral flow devices out of it. So what exactly is a lateral flow device? So it's a paper-based basically device in which initially it has a sample pad. So you let your samples flow through here. Now here are this is the conjugate pad in which you have signal reporters. Now, as your liquid that you are supplying passes through this, it comes across. So there are two lines. First of all, this is a test line. Test line is the one, basically this section will detect the target reacting molecules, and the other one is the control to verify the lateral flow system is working. This is the backing plate, and then this is membrane and absorption pad. So let us see what are the different, structural components of a lateral flow device. This schematic shows the components of the lateral flow device strip and how detection happens. So first of all, there is sample pad where the user applies the test sample. Suppose you have saliva, nasal swab, extract or blood. The liquid starts moving forward by capillary action. So it starts because obviously all of it is a paper based approach. So it starts moving by capillary action. Now you have a conjugate pad. What do you have on conjugate pad? It contains labeled biomolecules like antibodies, aptamers, or DNA probes, conjugated with visible tags. Example, gold nanoparticles, fluorescent dyes. So suppose it contains labeled biomolecules, जैसे कि antibodies or aptamers. What are aptamers? Aptamers basically are short sequences of DNA only. So you suppose here you have short sequences of DNA to which you have conjugated some fluorescent dye. So if some analyte is present in your sample, it will come and bind here. It will come and bind here, and when it will move, if the target analyte is present, it binds to these conjugates. Now there is test line coated with primary antibodies, which is specific to the analyte. Now it has some primary antibodies. When these molecules start moving, so this particular molecule will come and bind with its complementary antibody, and it will show some, that could depend what, if you are using a fluorescent dye, it will show fluorescence here. So specific to the analyte, captures the analyte conjugate complex, forms a visible control line, जिसे हम test line भी कहते हैं, we call it T line. Then there is a control line, this coated with secondary biomolecules that capture excess conjugates, ensures the test works properly. And then there is absorption pad, which wicks away fluid, maintaining flow. So let us visualize it more correctly. Suppose this is a lateral flow device which is being made for detecting Klebsiella pneumoniae. So there are different lateral flow devices which are used to detect different biomolecules or different basically I would say obviously yeah molecules that is associated with some kind of diseases. So in this case they are basically detecting Klebsiella pneumoniae. So here panel B example of a lateral flow device test results. Here the device is being used to detect Klebsiella pneumoniae. Okay, a pathogenic bacterium, which is a pathogenic bacterium. So the top strip buffer only negative control. So in this case, when Klebsiella pneumoniae infection is not there, so you would see a line is there which is only in the control sample. Means the test sample that you have added, Klebsiella pneumoniae is not present here. But in this case, you would see two lines. Okay, you would see two lines here. Means the sample that you have loaded in this particular tag is positive for Klebsiella pneumoniae, which is a pathogenic bacterium. So bottom strip, the both C and T line appears. The T line indicates binding of the bacterial target to the conjugated biomolecule. So it shows positive for Klebsiella pneumoniae. So by this, I just wanted to show how DNA-based nanotechnology are currently being used for different biomedical applications, for heavy metal detection, for basically biosensing, for disease detection by formulation of such lateral flow devices. Moving forward, another DNA-based approach is these deoxyribozymes. What are deoxyribozymes? These are also called as DNA zymes. So sequences catalytic activity like protein enzymes, but simpler. So there are certain DNA sequences which have catalytic activity which mimics basically how enzymes work. So they can cut RNA, bind ions, or they can also catalyze reactions. So what are the applications? Biosensors for detecting metal ions or toxins, gene silencing, and suppose you have RNA cleaving DNA zyme, a particular sequence. So what it can do, it can basically, where does my pointer go? Okay, just a second. Okay, so what it can do is this particular DNA enzyme, what it can do, it can cleave messenger RNA. What does messenger RNA do? Messenger RNA basically carries the information, and then when it basically it leads to translation, protein translation. So if it is cleaving the messenger RNA, obviously the information will not be translated, and there would be gene down regulation. So it can do, it can basically regulates gene down regulation, gene up regulation, and apart from that what it can do is suppose you are using a particular sequence, it is going and binding to a target, and if you have added some kind of fluorophore to your sequence, what would happen? It would give some kind of fluorescence which can be used for different biosensing applications. So what are you guys should know what exactly are deoxyribozymes. What are deoxyribozymes or DNA zymes and how does it function? So DNA zymes basically are a set of sequences which have catalytic activity and which can catalyze reactions, cut RNA, or perform different functions. Now, next thing is this is the next category. Okay, just a second. I hope I'm still clear. Just a second. Okay. So in the next section, what is happening is this is the next category. Um, Inorganic nanomaterials guided by DNA. So in the first section, we saw how DNA nanotechnology, how when DNA is being used alone and modified it when DNA only is a raw material. Now, in the second section, we will see how we incorporate DNA with different inorganic materials and use for different applications. So here DNA is used to arrange or functionalize non-biological materials, okay, for hybrid applications. Already we know what we are doing here is we are combining DNA, we are incorporating DNA with different inorganic materials for hybrid applications. That is why. So gold nanoparticles can be coated with DNA strands. They show unique optical properties, plasmon resonance. Okay. So Suppose how does this happen? Suppose gold nanoparticles are alone, or if suppose there are some DNA strands and you're recording some signals from that. Now, once you coat your gold nanoparticles with some DNA strands, it will go and bind. Suppose this is the site, this is the target site. This will go and bind at a particular target site. So when it go and binds to a particular target site, and this target site has gold nanoparticles in the close vicinity, so it shows some plasmon resonance which you can recall. So this is here DNA is basically used is being used for targeting and for going and attaching to a particular target, which leads to enhancement in the signal. Application: colorimetric biosensors change color when target binds. So again, they could be change in the plasmon resonance, or when it goes. Suppose your gold nanoparticles are usually red wine in color. Okay. Now when it is going and binding to a particular target, it might aggregate. So while aggregating, you could see a visible cue. It changes color and turns purple. It might turn purple. So colorimetric biochanges change color when target binds. So it might change color when it goes and bind to a particular target. Cancer treatment via photothermal therapy is one thing. Imaging probes. So DNA coated gold nanoparticles can hybridize with target DNA RNA sequences. Binding events change the state, aggregation, dispersion of nanoparticles, and there is measurable signal. Okay. So gold nanoparticles, which is a colorimetric sensor, it interact, analyze to have quantitative signal. Color changes, causes of aggregation. Then there are chemical sensors, and then there could be biological sensors as well, which leads to biological recognition elements, quantitative, semi-quantitative, analytical information. So enzyme-based or DNA-based assays you can do, or you can also use it for heavy metal detection. Now, two-dimensional nanosheets. Now, these are very important. Please understand what is happening here. So here, materials like graphene or molybdenum disulfide can bind with DNA. So do remember again, this is here, it is DNA as well as inorganic material, which is graphene and molybdenum disulfide. So DNA provides stability, selectivity, and programmability, right? So what are the applications? Energy storage devices, biosensors for DNA protein detection, and platforms for drug delivery. Now, suppose this example is there. Suppose this example is there. What is there? This is vanadium disulfide quantum dot. What you see here in blue, these are vanadium disulfide quantum dots to which we have attached tetracycline aptamers. What are aptamers? Aptamers are short, single-stranded DNA or RNA molecules that fold into a unique three-dimensional shape to bind a specific target, acting like antibodies, but with advantages such as smaller size, better stability, and easier chemical synthesis. So always remember, aptamers are short, single-stranded DNA or RNA molecules only. Now, we have attached aptamers, basically DNA sequences, with a vanadium disulfide quantum dot, and then impregnated that, basically immobilized that on the this molybdenum sheet, molybdenum disulfide nanosheets. Okay, so whenever in absence of tetracycline, so basically this particular test is being used to detect this molecule. What is this molecule? This is tetracycline. So when there is no tetracycline in the sample, so it will stay like this only. But if there is tetracycline here, this tetracycline aptamer will bind to these tetracycline molecules, and this vanadium disulfide quantum dots would be detached from the molybdenum disulfide nanosheets, and this would be detected. So what is happening in this particular system? A system composed of vanadium disulfide quantum dots and molybdenum disulfide nanosheets for use in aptamer-based fluorometric tetracycline assay was developed. The tetracycline aptamer was first immobilized on the VS2 quantum dots with a typical size of 3 nanometer. Can you imagine? These quantum dots are 3 nanometer in size, so small. So first of all, what they did is they immobilized these tetracycline aptamers on these vanadium disulfide quantum dots. After that, the blue fluorescence of the VS2 quantum dots with emission maxima at 448 nanometer was subsequently quenched by MOS2 nanosheets. So initially they had a blue fluorescence, but when they immobilized this aptamer plus vanadium disulfide quantum dots on the molybdenum disulfide sheath, their fluorescence was quenched in absence of tetracycline. Now, when tetracycline comes into the sample, comes in the scene, it binds with this. What happens is it is again detached from the molybdenum disulfide sheet and again shows blue fluorescence. So this is a turn on stage. This is a turn on condition. This is a turn off condition. So by this example, what I wanted to tell you, what I wanted to explain is how DNA can be incorporated with some inorganic materials and can be used for different applications. So here they formed these nanostructures for detection of this tetracycline. This tetracycline was being detected if the samples had tetracycline. Moving forward, DNA-based nanosheets for energy storage applications. Now, how can DNA-based nanosheets be used for energy storage applications? So, DNA nanosheets are thin, sheet-like structures formed by the self-assembly of DNA. We all know that. And they are increasingly used in energy storage applications because of their high surface area, because they can undergo self-assembly and form nanostructures. And we all know that nano-based structures have high surface area, and we can easily, because of the large surface area, we can functionalize those with chemical groups and ability to hybridize with conductive materials. So on their own, DNA nanosheets can bind and transport metal ions like lithium or sodium through their phosphate backbone, creating nanochannels that facilitate smooth ion movement. So this is the crux of the concept, that it creates DNA, these DNA nanosheets create nanochannels which facilitates smooth ion movement. There is movement of ions, and if there are proper movement of ions, obviously there is kind of like there is conductivity that is being given. When combined with materials such as graphene, metal oxides, or conductive polymers, they form hybrid nanocomposites with excellent conductivity and stability. So they provide excellent conductivity as well as stability. So that is why they are used for different energy storage applications. These DNA-based hybrid nanosheets serve as efficient electrode materials for lithium-ion and sodium-ion batteries, to remember, and as active layers in supercapacitors, enabling high charge storage, fast charging, discharging, and low cycle stability. So their biocompatibility, programmability, and eco-friendliness make them promising candidates for next generation green energy storage and flexible electronics. So this is how different DNA-based nanosheets are used for energy storage application. So whenever we are talking about application of a particular technology, of a particular approach method, we should always know how it is happening. Okay, if I say a simple statement that DNA-based nanosheets are used for energy storage application, and you just memorize it in your mind, okay, they are used for different energy storage applications. But apart from that, you should definitely know that how is it happening? Why? What is that special feature? What is that special quality in those DNA-based nanosheets? What is so special about their fabrication, about their structure, that they can be used as energy storage application? So you should know that DNA has a property of self-assembly and obviously large surface area. They can functionalize, they can form nanochannels and facilitate smooth movement of ions. So that is why they are used in different lithium ion and sodium ion batteries. So in addition to that, their biocompatibility, programmability, and eco-friendliness make them promising candidate for next generation green energy storage. So as we all know, it's a biomolecule, obviously. So green energy storage and flexible electronics. Now another method. Okay, so why I have put this particular example here is because you will understand a new method which we call FRET, Foster resonance energy transfer, or it is sometimes also called as fluorescence resonance energy transfer. So what happens in FRET? Suppose DNA combined with a fluorescent nanoparticle, suppose quantum dots or upconversion nanoparticles. So suppose this is your quantum dot, okay, and suppose this particular quantum dot gives you cyan color, okay. This particular quantum dot, when it is alone, it gives cyan color. Now, suppose you impregnate it with different functionalizing agents or DNA, and when it encounters a target DNA, okay, so what happens is when this target DNA comes in close proximity to this quantum dot, there is FRET emission coming. So in that case, the fluorescence of this particular quantum dot goes gets transferred to this the fluorophore attached to this target molecule, and instead of cyan color it starts emitting the emission given by this one. Now let us understand it more clearly. So this image shows how quantum dots coated with DNA strands are used for DNA sensing via FRET. Normally, when excited by light, quantum dots emit their own fluorescence. Just now I told you when quantum dots are alone, they emit their own fluorescence. Now, when target DNA binds to the complementary DNA on the quantum dot, fluorophore labeled DNA strands, these fluorophore labeled DNA strands come in close proximity, enabling energy transfer from the quantum dot to the fluorophore. So what is happening in FRET is there is energy transfer from the quantum dots to the fluorophore. This results in a new emission signal, which we call as FRET emission. So there is energy transfer from this quantum dot to this fluorophore, indicating the presence of the target DNA. This mechanism allows highly sensitive and specific detection of DNA sequences. Let us understand what exactly is happening in FRET. Suppose there are two proteins, protein A and protein B. Now, in this case, suppose these two proteins are there, but they are well separated. Okay, they are well separated at d distance. This is protein A. This protein A, we have tagged it with cyan fluorescence protein, and protein B we have tagged with yellow fluorescence protein. So it will give blue emission and this one will give yellow emission alone. Now we all know when we're talking about cyan fluorescent protein or yellow fluorescent protein, so the excitation wavelength would be different in both of them. Suppose if this one gets excited by 430 nanometer, the excitation wavelength for this one would be different. Suppose if we are exciting the sample with 430 nanometer, we'll record emission, CFP emission at 475 nanometer. Okay. Now in next case, when protein A and protein B interacts, see this situation. Here, it is not separated, it is not well separated, it is in very close proximity. Protein A and protein B, when it comes in very close quantity, uh, close proximity, sorry, not quantity, when it comes in close proximity and we excite it with 430 nanometer of light, we excite it with 430 nanometers of light. So the emission from basically the energy from this gets transferred. That is why we are using the term this Foster resonance energy transfer. It gets transferred to this yellow fluorescent protein and we see basically YFP emission. So here the emission there would be cyan emission, basically blue emission. Here yellow fluorescent protein, we'll see yellow emission. So this is Foster resonance energy transfer. This is the whole concept. So for it to happen, what is the necessary condition? That the two interacting moieties, like in this case, the two protein molecules, protein A and protein B, shall be in close proximity. Now, we'll also see how this particular concept, how this particular theory is being used to make different bio nano machines. There was this topic in your this bio nano machines in which BZ motor, how it is used, how the same concept is used for synthesis of this BZ motor. So in Foster resonance energy transfer, cyan fluorescent protein and yellow fluorescent protein are commonly used as a donor acceptor pair. So here it is donor because it is donor and it is this one is acceptor. So CFP, which is cyan fluorescent protein, when excited with a specific wavelength. CFP emits cyan fluorescence. So yellow fluorescent protein, which is acceptor, is close enough. Always remember 1 to 10 nanometer. If the two moieties lies in close proximity, that is 1 to 10 nanometer. If the distance increases, FLET will not occur. So if the distance is 1 to 10 nanometer, the energy from the excited CFP is non-radiatively transferred to yellow fluorescent protein, which then emits yellow fluorescence, This means that if CFP and YFP are brought into proximity, for example, via protein-protein interaction or DNA hybridization, you will see reduced CFP emission and increased YFP emission. By measuring this change, researchers can study molecular interactions. So how is it useful? Suppose I want to check if the target, if this molecule is interacting with this one, and we utilize this, we tag it with CFP and we tag it with YFP. So if this particular molecule is properly interacting with this one and comes in close proximity, so there would be more yellow fluorescence. But if it is not interacting and it is staying away from this particular molecule, there would be cyan fluorescence. So this is how this concept of FRET is being utilized for different applications. So always remember. Now, one thing you should always remember, that whenever we are talking about self-assembly, always remember that self-assembly follows a bottom-up approach. Suppose see this one. There is self-assembly of CDT nanoparticles into spherical and uniform supraparticles, exhibiting a significant reduction in polydispersity. Then agglomeration of gold nanoparticles at high and low hemoglobin concentration, as you can see. Okay, these are gold nanoparticles, these are hemoglobin. So in case of high agglomeration of gold nanoparticles at high left and low right hemoglobin. And the third figure here shows growth stages of the high temperature assembly of ZNSE nanoparticles, leading to couples of colloidal nanorods, how they are being formed. So all this self-assembly approach basically follows bottom-up approach. Now let us come to solving the assignment quiz that was given to you, that you have to submit, I guess, by 3rd of September. So let us discuss some of the questions. Just give me 2 minutes. I want to drink water. Okay. So the first question that was asked to you in your second week's assignment quiz, select the correct list of lipids. So there was a section in your in the different videos that was uploaded, lipid nanotechnology. So in that lipid nanotechnology, you guys were told what are the different materials that are used for forming lipid-based nanostructures, how they are useful for different sensing, drug delivery applications, and all of those things. So in this particular question, you have to select the correct list of lipids. Okay. So first one, it is dextrose. Obviously, it will not be there. So I guess if you guys want to answer, if some of you knows the answer, please, or I'll just wait for basically 10 seconds and then I'll tell you the answer, and why, and apart from telling you the answer, I'll also discuss like why is it so. So basically this one is to help you guys to better solve your assignment quiz questions, and to check as well that whatever you have studied, or if you have missed some questions, or if you're not able to solve it correct. So the answer is the third one. Nice, very good. So, see, whatever questions that are being given to you, that basically is based on the course content that is already uploaded on your NPTEL website. But apart from, just because, see, if you just want the answer, if you are searching it on Google, you might find the answer. But apart from knowing the correct option, all of you should know why that option is correct. Why not option 1? Why not option 2 or option 4? So, as we all know, these are all lipid or lipid related molecules. So, fatty acids, we all know, blocks of many lipids, building blocks of many lipids. Glycerol, backbone of triglycerides and phospholipids. Cholesterols, waxes, triglycerols. These are all some form of lipids or fat only. The other options include sugar. So dextrose pele I cut out because it's a sugar. So then amino acids, glycine. So obviously this option would not be there. Then there are lipases. Lipase basically these are enzyme that degrades lipids. So lipase will already not be the answer. So you can follow this remove out kind of approach to come to the correct answer. Or it's very obvious also that this would be the correct answer. Now moving forward, this was your second question. Just now we talked about what is self-assembly and what are the features of self-assembly, what happens in self-assembly. So please do this one. Please try attempting this particular question. I'll wait for 10 seconds. Anyone? Okay, so those who have joined now, we are solving the week 2 assignment quiz. So I just want your participation. If you are listening to the lecture, listening to the session, uh, please let me know what you think. What shall be the answer? So the question is, which of the following is not the characteristic feature of self-assembly? We know self-assembly is uh, basically a spontaneous process. Obviously, because these are, suppose we talked about simple polymeric, how do we form polymeric nanoparticles using self-assembly? So these are there. Obviously, we add some reducing agent or some salt in it, which induces its self-assembly. It starts self-assembling. So this process obviously is spontaneous in nature. It's not like very controlled thing because it's not also possible that whatever particles are forming are exactly of same sizes. So if something is like that, some folding might be there, some might also be some smaller. So it's not like extremely controlled. Now self-healing capability, obviously it will be there. Now making ensembles of nanostructure, obviously we are forming nanoparticles here. So this one is also correct, but it tends to create defects. Actually, it does not create defects. And when we're talking about self-assembly, it's not just limited to nanoparticles. We also studied about DNA origami. So in DNA origami, we saw that the structures, suppose some this kind of structures are being formed. So it was very highly ordered. So no there was no defect kind of thing. So the correct answer would be what I think I have pointed. Just let me just a second. I have told you the correct thing only, but I have marked it up. The correct option is this, and same thing is written here as well. Sorry for that. So, not the characteristics. So, self assembly is typically characterized by being a by being a spontaneous process. It has self healing capacity and it enables formation of ordered ensembles of nanostructures. So, and it is designed to minimize defects, not create them. So, self assembly that we see, the DNA origami structure formation, highly ordered structures. So, it is used to minimize defects, not create them. So, tends to create defects is not a characteristic feature. Now moving on to the next question. The next question was molecular model of lipid-like peptidases, which is A6D, can be read as. Now, to attempt this question, there are no other options, there are no common senses. You should definitely know what exactly is lipid-like peptidases. What does this mean? So this has been taught to you in the content lecture that is available on the NPTEL website. But if you guys are not able to solve this one, obviously we'll discuss what are lipid-like peptidases. What does this mean? What is this meant by A? What does A denote? What does C6 denote? And what does D denote? Anyone who knows what does these particular letters denote here? So based on the options that are available, you can see either it could be A could signify either alanine or arginine. Okay. So do you guys like remember or do you guys know that there is like nomenclature given like alanine, if it is alanine amino acid, then you single letter nomenclature, we name it as A. So D basically is aspartic acid. Okay. So to attempt this question, you should have like simple biochemistry that you study amino acids. If you guys have studied this topic, amino acids, I guess you would be able to understand better what exactly is happening. So, when we talk about amino acids, obviously there are um, positively charged, negatively charged, and then apart from that, uh, you know, hydrophilic ends, hydrophobic ends, all of those are present. But obviously, I know this is a little tricky question, and let us discuss. So the correct option here is the first one. Peptides have six hydrophobic alanine residues. So A, six, right? So six alanine residues, six hydrophobic alanine residues from the N terminus, followed by negatively charged aspartic acid residue. So D here stands for aspartic acid, which is negatively charged. Okay, so this is the correct answer. But let us see what does it mean. So these are different lipid-like peptides that are available. So space-filling molecular models of surfactant peptide. So the first one is A6D. This one is V6D. So what is the difference? Here the hydrophobic part is constituted of six alanine molecules, and in V6D there are different valine molecules. Valine basically amino acid, and L-leucine, L6D2 constitute the hydrophobic tails with increasing hydrophobicity. Correct? Then green carbons, red oxygen, blue nitrogen. Here it is they are showing that which color spheres represent what, and each peptide is 2 to 3 nanometers. Can you imagine? Each of these peptides are only 2 to 3 nanometers in length, similar to biological phospholipids. So what would these do? Just now you were told that these are hydrophobic. They have hydrophobic ends. Okay. So what would happen? Suppose if we want to use it for targeting purpose, if we want to use it for different targeting purpose, what will it do? It will go and interact basically with the cell membrane because that is also hydrophobic. So it would increase its basically I would say permeation or entry into the cell. So for targeting, it can be used, and obviously you can functionalize these with some drug molecules that you want to deliver or for different applications. Now, the next question, the effect of salt concentration in solution on DNA structures are defined as. I guess this one you guys would be able to do. Anyone? I'll wait for 10 seconds and then I'll tell you the answer. So what exactly happens? So what exactly happens when you add uh, salt salt into the solution? Effect of salt concentration in solution on DNA structures are defined as, okay. Okay, so the correct option would be the third one. Why? So there would be decomposition of DNA structure in low salt concentration. So DNA has a negatively charged phosphate backbone, and salts such as sodium plus, magnesium plus, etc. stabilizes DNA by shielding these repulsive negative charges. So what happens in these two different conditions? At low salt concentration, insufficient charge shielding. So there would be electrostatic repulsion, and because of electrostatic repulsion, DNA denaturation or decomposition would occur. But in case of high salt concentration, it stabilizes DNA. So that is why just now we read that we use magnesium 2 plus ions to stabilize them, right? There shall be presence of magnesium 2 plus ions for stabilization. So it stabilizes DNA. But again, there is an you cannot say that high salt concentration is effective for DNA stabilization. So there is a term called optimum salt concentration. So there is a term called optimum salt concentration that should be there, because if it will be low, then also it will be destabilized. If it will be high, then also DNA denaturation and destabilization would occur. So there shall be there is a range, I could say there is a proper window in which it is stable. So at high salt concentration stabilizes DNA, but if extremely high, it can cause aggregation or decomposition. Now to understand this, what is happening exactly at low salt concentration, uh, you should know the concept of charge shielding. Suppose these are the DNA, these are the salt molecules, suppose, and you have your DNA structures here in presence of these salt salt molecules. Okay, pardon me for the such a bad drawing. I was just trying you to explain. So there is insufficient charge shielding. I mean, if there are molecules in between DNA structures, these salt molecules with some charges. So in case of low concentration, what would happen? Low salt concentration, these molecules would come close to each other, okay, which will lead to DNA decomposition. But in case of high salt concentration, what happens is, when we were discussing about basically dynamic scattering in zeta potential, what happens, why higher charge is considered stable? Because if they have significant charges, similar significant charges, suppose positive, positive, what would happen? It will repel each other. So when it will repel each other, it will not come together and aggregate. So that is why it is said that at high salt concentration it stabilizes DNA, but at low salt concentration it will start coming together, and because of the electrostatic repulsion would be low, insufficient charge shielding, basically electrostatic repulsion and DNA denaturation or decomposition would occur. So basically this all concept is based on charge shielding. If you will just read once or twice, you will know. Okay, so I have already, told the answer in this one, but it's okay because this one is actually a little typical one. Because if you guys don't know about bio nano machines, what is BZ transition? Anyone knows what exactly is happening in this BZ transition? So simply BZ transition that is currently being talked here in this particular question is transformation of B form of DNA to zDNA. Okay. So this particular concept is being utilized here. So choose the correct statement. Okay. The BZ transition is based on the change in the nitrogenous base pair sequence in the presence of helicase enzyme. BZ transition, एक second. Okay. So, BZ transition occurs by changing the ionic strength of medium. Second option, Fred used to study the energy transfer between two light-sensitive molecules. We have just now discussed Fred. We'll see what is happening in this particular question. Fluorescence quenching is observed in zDNA form of BZ DNA motor. So the correct option obviously is this one, but why this one is correct? Because uh, why these options are correct, we'll discuss one by one. What is FRET? How is FRET regulating this BZ transition motor and everything? But let us dive into what concepts basically guides this kind of process. So to understand that, first let us understand what is BZ transition in DNA. So DNA, as we all know, normally exists in the B form under physiological conditions. So under physiological conditions, our DNA mostly exists in B form, hydrated right-handed helix. As you can see, bDNA right-handed helix. But under certain conditions, it can switch to the Z form, and this shift that we call zDNA, and this shift is called BZ transition. We call this BZ transition. Now bDNA कॉमन फिजियोलॉजिकल कंडीशंस में ये पाया जाता है. राइट हैंडेड हेलिक्स, major and minor groups are well defined, stable in normal salt concentration and physiological conditions. Now, what are the differences observed in zDNA in terms of characteristics when compared to bDNA? This is alternatively left-handed helix. Okay, more zigzag shaped backbone, hence Z, has 12 base pairs per turn, occurs in DNA regions rich in alternating purine, means GC rich sequences जो होते हैं ये वहां पे पाया जाएगा. These will be uh, these will be found in GC rich sequences. Okay, stabilized by high salt concentration or certain chemical modifications like methylation or of cytosine. PZ transition refers to the reversible shift from bDNA to zDNA under suitable conditions. So, if there is high salt concentration, if there is negative supercoiling, there is some DNA methylation or binding of zDNA binding proteins, it will lead to transformation of bDNA to zDNA. It is functionally important because zDNA forms transiently near active genes. So basically, it helps in gene regulation. So from this particular slide, what needs to be understood is what exactly is meant by B to Z transition. What are the characteristic differences in zDNA when compared to bDNA and under what conditions it is happening. Now we understood what is BZ transition in DNA. Now, how it is being used, because all this concept, this question basically comes from your lecture slide, which are bio-nano machines. So what are bio-nano machines? These are molecular scale devices made of biological molecules like proteins, DNA, RNA that perform specific mechanical or chemical tasks inside living systems or in synthetic environment. They operate at the nanometer scale, 1 to 100 nanometers, and are often inspired by or directly taken from natural biological systems. So, if you remember, there was one concept that we studied in the last week, biomimicry. So, in biomimicry, we saw like shark skin based swimsuit. Why? Because there are the tubercles that are present, they cut out waves and helps the swimmer. So all those are bio-inspired designs, means what is already present in nature and based on those designs, the nature-inspired designs. So same bio-nano machines are also the same. So bio-nano machines are molecular robots of nature or bio-inspired designs. So what are the key features of bio-nano machines? Built from biomolecules like proteins, DNA, RNA, lipid. We'll discuss about this one, BZ motor. Operate through conformational changes. Just now we discussed how the conformational changes are happening, B to Z transition are happening. Capable of self-assembly and precise function at the molecular level. So it performs tasks like transport, energy conversion, sensing, or catalysis. So these are some of the features of bio-nano machines. We'll discuss one example, although these are natural bio-nano machines. What is meant by natural bio-nano machines? means which are already found in our body. Okay, natural, already found in nature. These are not engineered one, or these are not synthetic ones that we have synthesized by getting inspired from their machinery. So these are the examples. These are some examples of natural bionanomachines. So one of them is ATP synthase, molecular motor that synthesizes ATP. Okay. Ribosomes, we all know what function ribosomes do. It translates messenger RNA into proteins. We all know how does it work, how it's how it moves through the and it translates the protein. Then the other one is kinesin and dynein, means motor proteins that walk along motor proteins that walk along microtubules to transport cargo inside cells. This is the example that I have added here. Now let us understand. So both kinesin and dynein. What are these? What is kinesin and what is dynein? These are motor proteins. What does motor proteins do? These are tiny bionanomachines inside cells that move. Just now we said these are motor proteins. Motor proteins means it will move. So it will move along microtubules, which are part of the cytoskeleton. They carry cargo such as vesicles, organelles. Suppose there are some vesicles or organelles or proteins that needs to be transported from one part to other part. So what does kinesin and dynein do? These are motor proteins and they move along microtubules and transports cargo to different parts of the cell, almost like delivery trucks on cellular highways. So these are the cellular highways, and these are the delivery trucks, this kinin and dynein, and they transports cargo. So what is the structure looks like? Kinesin linear motor on microtubules. Kinesin consists of light and heavy chains. Okay, the light chain binds to kinesin receptor. So this one, this one is the kinesin light chain, and it binds with kinesin receptor. Okay. Then the light chain binds to kinesin receptor on vesicles that are to be transported. So these vesicles need to be transported. This is the cargo. The heavy chains each include motor domains that use ATP as energy to move kinesin plus the attached cargo along the surface. So this blue colored region, basically this forms the heavy chain, kinesin heavy chain. So this kinesin heavy chain is composed of different parts: C-terminal tail, coil domain, motor domain. Okay. So what does it do? It uses kinesin heavy chain uses ATP for the movement as energy to move kinesin plus the attached cargo. It moves whole of it along the surface of a microtubule. So just now we saw this is a naturally occurring phenomenon. Naturally occurring bionanomachines. So based on this, inspired from this, there are different experiments, different approaches, different methods being developed and used as this bionanomachines. One of the examples include this. So there are different artificial or engineered bionanomachines available. One of it is DNA nanorobots that can deliver drugs, synthetic molecular motors powered by light or chemical fuels, protein-based nano devices designed for biosensing. So one of it is BZ motor. Now, what is a BZ motor? A BZ motor is a type of synthetic DNA nanomachine that harnesses the B to Z transition as a source of motion. Okay, so how does it function? The motor is built from a DNA segment rich in alternating CG sequences. Okay, so this is built, this DNA motor is built from DNA segments which is rich in C and G sequences. Okay, when environmental conditions change, like salt concentration, supercoiling. Suppose under some conditions there is supercoiling happening, or if you have protein ligand kind of thing, so your there is binding of your DNA sequences with some complementary proteins or ligands. So obviously there would be some changes in its conformation. So the DNA switches between B form and Z form. This structural transition involves a conformational flip in the DNA helix, which produces mechanical motion at the nanoscale. Now, this one is very important. Okay, See this diagram. So what is happening here is, suppose B to Z transition and Z to B transition. Now what happens here is, see, and how fret is coming in picture. Ways of BZ DNA motor works in biosensing. So optical biosensing is that fluorescent dyes or fret pairs are attached to DNA. So fluorescent dyes or fret pairs are attached to DNA here. Okay. Just now we studied fret, that when two different basically fluorophore tagged pairs are attached, and if they are in close proximity of not here, but if they are in close proximity 1 to 10 nanometer, there is Foster resonance energy transfer, and we see different fluorescence. So when there is change in conformation, whenever there is change in conformation, so whenever there is a conformational change in DNA, suppose Z to B or B to Z, there is also obviously there will be change in the position of these fluorophores. So whenever there is a change in the position of these fluorophores, obviously the fluorescence signal that will be detected would be different depending on their proximity, their closeness, how close they are lying to each other, and if fret is happening. So this is how it is being exploited. This is how it is used. How this BZ DNA motors are used for different applications. One of it is biosensing. So optical biosensing, what is happening in optical biosensing when DNA flips from B to Z? When DNA flips from B to Z, the distance orientation between dyes change, measurable change in fluorescence. What happens in electrochemical biosensing? DNA is immobilized on an electrode. BZ transition changes the electrical charge distribution. So here the visible cue is fluorescence. Okay. So whenever there will be a change in conformation, you would see the difference in fluorescence intensity or fluorescence color. So here, whenever there would be a change in conformation, you would see a change in the current or voltage. The current or voltage would change. So this is how this BZ DNA motor or bio nano machine can be used for different applications. So as the and how fret governs it. So that is why based on these conditions, the option that question was done. Moving forward, sixth question. Okay, so I have already, so let us just discuss the answers and then uh, the basically the correct answers because there is very less time left. So let's discuss it. So the first statement is proteins, ions, DNA can be used as nanomolecular communication carrier in living organism. And the other statement B is communication distance in molecular communication is measured in nano and microscale. So obviously both of the options are correct. The first one is proteins, ions, DNA can be used as nanomolecular communication carrier. Obviously we tag our inorganic nanoparticles with some peptides, with some targeting DNA, I mean aptamers based in living organisms. So they can be used as nanomolecular communication carrier. Apart from that, the communication distance shall be nano and microscale because if it will be too distant, obviously there will be no proper targeting happening. So option A, both A and B are correct. Next question was non-solubility of carbon nanotubes in aqueous medium can be resolved by. So there are different approaches used to make the carbon nanotube soluble. Usually in its raw form, carbon nanotubes are non-soluble. These are non these are non-water soluble. Okay. So there are different approaches. There are different approaches which are currently followed. There are different approaches which are currently followed to make it soluble. So carbon nanotubes, that video lecture you guys would have already seen it. So now let us just quickly revise what exactly are carbon nanotubes, how they are formed, and what are the different approaches that are currently available to make the carbon nanotubes soluble in water. So first one is obviously functionalization of carbon nanotubes by adsorption, by electrostatic interaction, by covalent bonding. So these are the approaches. So non-solubility of carbon nanotubes in aqueous medium can be resolved by doing all those things. So addressing the limitation of carbon nanotubes. One of the biggest challenges with carbon nanotubes is their poor solubility and dispersibility in water and organic solvent. Okay, so see, in on the NPTEL website, you would find the pre-recorded video lectures by the faculty from IT, I guess IT Roorkee. Professor P. Gopinath has already uploaded the video lectures of different topics, lipid nanotechnology or bio nano machines, DNA nanotechnology, everything. Those are pre-recorded lectures. So apart from those pre-recorded lectures and the content material that is available on your NPTEL website, there currently there are three live sessions happening by different course members. Like one is taken by me and other two people. There are two other people also who take live session on these materials. These live sessions basically are designed for you people. Basically, if you guys have any doubt or if you need additional course material or if you want to understand some topics more clearly or if those topics are not clear to you or not understood by you, you can ask those things here. Apart from that, this particular session is also designed to discuss your assignment questions and some sample questions as well, if possible. So this is basically doubt solving kind of session in which apart from discussing the assignment questions and some sample questions, we are also there to address if you have any doubts in any of the topics, uh, any of the concepts, any of the, uh, if the lecture slides that are already uploaded on the NPTEL website, if while studying those lecture slides, those content material, if you have any doubts related to any topic, you can ask us here. And one thing I would like to tell you people, suppose week 3 content is already uploaded on the NPTEL website. And now, while studying the week 3 content, if there are certain topics that you guys want to revisit again, if you guys want to discuss again, one thing you can do when I'll upload this lecture on YouTube, you can comment which topics you would want to discuss in the next session. So, in that case, what I can do, I can focus more on those topics for you people to understand those topics better. So it all depends on how better you are interacting with me, how better you are telling me your problems or the topics that you are not able to understand properly. So I will again properly revisit those topics, discuss those topics again, and I will try to explain those topics in a more simpler manner or in a bit detail so that you understand that. Now coming again to it. So these are the different approaches that we follow to basically make carbon nanotubes soluble in water. You see, these are not soluble, these are aggregating, but here it is soluble. So covalent functionalization. So what do we do in covalent functionalization? We attach groups like COH group, OH polymers to CNT surface. They are stable but may affect properties. Sometimes the properties may be affected. Non-covalent functionalization, in this case, we use surfactants, polymers, or biomolecules to wrap CNTs. So, in this case, the properties of carbon nanotubes are preserved. Endohedral functionalization fills CNT interior with molecules for dispersion. Solvent dispersion use solvents like DMF, dimethyl formamide, and MP plus sonication to separate CNTs. So these are the different approaches that are currently available to solubilize CNTs. So was the eighth question. So this question is basically based on the methods, the different synthesis methods of carbon nanotubes. So carbon nanotubes basically can be synthesized via different methods. And based on which synthesis method you use, the type of carbon nanotubes that are synthesized in that particular method differs. What are the different methods? These are the different methods for carbon nanotube synthesis. CNTs can be made by several methods, and the most common are arc discharge method, laser ablation, chemical vapor deposition, and other methods. So let us discuss two of them. Like in arc discharge method, there are two graphite electrodes, cathode and anode, and which are two graphite electrodes are vaporized by a high current 50 to 100 ampere in an inert gas atmosphere. Always remember this. There is some inert gas atmosphere. It produces high quality carbon nanotubes but with impurities, because obviously the electrodes are made up of graphite. So obviously there are some impurities. Now, when we are using laser ablation, this laser beam is there, this is graphite target, and once laser beam falls on it, it will eject out basically uh, some nanomaterials, which is again cold, and then these carbon nanotubes are formed. So, high power laser pulses onto a graphite target at high temperature in an inert gas atmosphere. Carbon vapors that are produced, it cools and forms nanotubes, yields uniform, high quality of CNTs, but it is expensive and low scalability. Now, another method which is used, it has most scalability. Hydrocarbon gases are decomposed at high temperature over metal catalysts like iron, nickel, cobalt. Carbon atoms deposit and grow into nanotubes. So this is the most scalable one and widely used method for industrial production. So these are different methods of synthesis of carbon nanotubes. If we'll go in detail, you would understand why the carbon nanotubes synthesized in all these approaches differs. So these are the characteristics. Like this is a single volt carbon nanotube. This is basically double volt, and these are multi-volt carbon nanotube. One of the very important characteristic of carbon nanotubes is it is extremely strong, but at the same time it is lightweight. So it is lightweight but stronger than steel. So you can imagine electrical, it can be metallic or semi-conducting. It has excellent thermal conductivity, useful for heat dissipation, and it has obviously because it is a nanostructure. Okay, nanostructure, so it has high surface area, so it can be chemically modifiable, can be functionalized with biomolecules, polymers, metals, and stable, but can be oxidized for solubility and functionalization. So ninth question is, lipid like peptides can work as similar to. All these options would obviously be wrong because all these are enzymes. Okay, and lipid-like peptides are also used for drug delivery applications. Obviously, it will not be enzymes. So lipid-like peptides are synthetic amphiphilic molecules. Amphiphilic matlab it will have both hydrophobic and hydrophilic parts in it. This is the meaning of amphiphilic. Similar to lipids and detergents, like chemical surfactants would be the correct answer. So they have the ability to self-assemble into structures like micelles and vesicles and are crucial for solubilizing and stabilizing integral proteins outside of their natural lipid environment. So two important things are there. There are different lipid-based nanoparticles. One of it is solid lipid nanoparticles, and it is also there in your course content as well. So solid lipid nanoparticles basically are submicron colloidal carriers 500 to 1000 nanometers made from physiological lipids. One very important, I would say, characteristic is they are considered the next generation of drug delivery systems, combining the advantages of polymeric nanoparticles, emulsions, and liposomes. Now, one important thing is it can encapsulate both hydrophilic and hydrophobic drugs, controlled and sustained drug release, protection of labile drugs, and these are biocompatible and biodegradable. So as you can see, this is a surfactant layer. These are active principles. Active principle means you can even load your, suppose you are using it for drug delivery application. So you can load your drug here. This is lipid. So this is solid lipid nanoparticles. Okay. So solid lipid core, which is made of triglycerides, fatty acids, fats or anything. Then this is basically stabilized by or surrounded by a surfactant, which could be lecithin, polysorbate, bile salts. So basically, a layer of surfactants helps in better dispersion in the aqueous phase. Then drug molecules that you are loading in it. Now, another is lipid polymer hybrid nanoparticles. So, it's not nowadays the approach is they are not using any nanoparticles like only lipid based, only polymer based. So, they are combining benefits of both of them and then making a suitable delivery carrier. So, lipid polymer hybrid nanoparticles are core shell nanocarriers that combine the advantages of both polymeric and lipid based nanoparticles. So, it is composed of a polymer core, a lipid shell, and a peg outer layer basically for stability, so that it does, so the whatever we are loading in it, it does not leaches out. It provides a protective covering. So this is here, so this is the polymeric core, this is the lipid shell, which will basically increase the hydrophobicity and penetration in the membrane, hydrophobic tail, hydrophilic head. So advantages, what are the advantages? High drug loading, biocompatible, suitable for both hydrophilic and hydrophobic drugs, and controlled release plus targeted delivery is possible. Applications, obviously, uh, cancer therapy and imaging and diagnostics. You can also load some fluorescent compound in it and use it for imaging as well. So this is the last question. This one is a very simple one. So DNA denaturation occurs by what would be the correct reason? As we all know, what when does DNA denaturation happens? So obviously whenever we heat it, so at a particular temperature, so on heating, the hydrogen bonds between purines and pyrimidines breaks. So the correct option is the third one. Just a second. Okay, so that's all for today's class. This lecture would be uploaded on YouTube. So if you guys want to again revisit the lecture, you can do it from there. And in addition to that, the slides would also be uploaded in the Google Drive link. So if you want the content, you can access it from here. So in the next live session, if you guys have any specific doubts, specific questions related to any of the topics, please feel free to write in the comment box of the YouTube video, under YouTube video that will be uploaded, and I shall definitely discuss those topics if you guys have any doubts with some specific topics. So that's all for today's class. Thank you very much.